Okay, so we are back to our um, enzyme kinetics, and we're looking at the uh, transition state theory in terms of how enzymes work. And this is what I kind of did on the whiteboard earlier. We said that in a regular reaction of S to P, that you're starting at an energy level here, the product is of lower concentration, and then the enzyme helps lower the activation energy, and a decrease in the activation energy, a small decrease in the activation energy causes a large decrease in a large increase in the reaction rate or the speed at which the reaction goes up. Doesn't change the delta G at all. You can see that in this reaction coordinate plot, but it does change the speed in which that reaction um, occurs. So this is a kind of a theoretical model of how that occurs. This uses the so-called stickcase model. Here, the reaction vessel P is the substrate, in this case a stick, that goes to that gets broken. And I imagine that the game of pencils that I used to play in elementary school, kind of in that way. The transition state here represents the peak of this energy. So it's the highest point in the delta G here, and it is the shape of the structure in that peak. For this stick, it will be when the stick is bent, but not quite, but not broken yet. All right. And the transition state also is the point or the form of the structure in which you have an exactly equivalent likelihood of either going back to substrate or forward to product. So if you have an enzyme that is complementary to the substrate alone, then what happens? You have this huge protein that wraps itself around the substrate. Well, then what? Now you've actually made it harder to go to P because you have to exert more force on this stick in order to bend it because you have this massive thing that's surrounding it. And so that's shown in the reaction coordinate plot here. If the enzyme is complementary to the substrate alone, then you actually stabilize the enzyme substrate complex, right? And so this is of lower energy, and you've just made your job much harder because now you have to put that much more energy into the system in order to get up to this transition state. So enzymes are not designed to complement the substrate. And we know this because when we looked at KDs in the previous chapter, dissociation rates, we saw that enzyme substrate interactions typically have higher KDs than protein-protein interactions or protein ligand interactions or things of that nature, right? So we know that the enzymes aren't built to specifically recognize the substrate. Instead, the transition state theory says that the active site of the enzyme is actually built to encourage the development of the transition state. And so here, there are certainly contacts to grab on to the substrate, but the active site is actually developed to push this substrate into the transition state so that it increases the rate of the reaction. So in this case, these are metal along this that drives this transition state and then allows us to go forward. So here, in this case, the enzyme substrate complex is at the same relative energy as the substrate, but the activation energy is much lower than the activation energy in the original. Again, the product is still the product. It's exactly the same as it was before. So there's no change in the total delta G of the system. The only change that the enzyme does is affect the activation energy. So if that is the case, then there should be a structure that binds to the active site better than the substrate. Remember, transition states or chemical intermediates are not stable structures. So these aren't things that are around for any appreciable length of time. So the field of medicine deals in many cases with um, what are called transition state analogs. So an analogs is an approximation of something. And so these are things that look like the putative transition site that the enzyme is trying to um, drive. So these are stable molecules. They are molecules that exist, and they should bind to the active site. TSAs should bind to the active site of an enzyme better than the substrate. And so here's one. This is an adenosine to inosine transition. The proposed transition site, and you guys are doing this in organic chemistry now, or you have done it if you've taken it, is this structure. This is an unstable structure. You would not see that. But here's an analog over here, and let's see, instead of having this amine group here, there's a hydrogen, but you have the hydroxyl over there, you have the nitrogen over there. So it mimics the look of what we uh, modeled the transition site to be. And you can test whether you have a good transition state analog by seeing whether it has a lower KD than the substrate, right? Because that tells you that the enzyme might be using that particular model. So this is a way in which you can come up with a mechanism of action, and you can test it by designing analogs or stable analogs to the actual active site and determining whether you dramatically increase the binding affinity. This is how modern medicine works nowadays in many ways. You look for things that block enzyme function or and allow a body to work effectively. So, in the enzymes work in a couple of different ways, and since they're proteins, they all use protein the chemistry of the amino acids. And we should kind of keep in mind, and we've seen this all before, but you know, the amino acids have various functionality depending on their R groups. So some of them have ionizable hydrogens, right? Some of them are polar, um, or charged, right? And all of those causes a difference in the chemistry that's associated with the compounds. Uh, the ones that have ionizable hydrogens, histidine, serine, in certain contexts, aspartic acid, glutamic acid, those are very useful in the active sites of enzymes. And so there are three ways that we're going to talk about in terms of enzyme dynamics. There's acid-base catalysis, right? So that obviously involves the transfer of a proton, right? Um, so that you use either one of these ionizable hydrogen residues in order to drive a reaction. And so here's an enzyme, and here is a proton here from this histidine, and this histidine, um, well, here's one in which this histidine is acting as a base, this one's acting as an acid. They are either withdrawing protons or picking up protons to allow for the chemical change um, to occur. There are two types of acid-base catalysis. There is general acid-base catalysis and specific. So specific involves water. So the way I think of it is it's specific because there's one thing that has to be involved. The proton has to come from water. Right? General is basically everything else. So if the acid is an amino acid or the base is an amino acid, it's general acid-base catalysis. If it's specific, it always involves water. We're going to see later on when we look at chymotrypsin that it uses both general and specific acid-base catalysis for it to do what it do. Another mechanism is covalent catalysis, and like the name implies, it involves forming a temporary covalent bond um, in the mechanism. Remember, covalent bonds tend to be very stable, right? And they usually are indicative of irreversible reactions. So this is one in which it forms a temporary reaction. And the last one is metal ion catalysis, in which you use a metal cofactor in order to drive the reaction. And we will see, we're going to kind of use a prototypical example of these reactions of chymotrypsin, but we'll see how they work in the next series. So I think I'm going to stop here, and then we're going to look at an actual chemical enzyme mechanism, and we'll use a prototypical example of chymotrypsin in order to look at that. All right.